This is Agriculture Today, and I'm Samantha Bennett with the K-State Radio Network. Ahead of us on this Tuesday's program, University of Missouri Curator's Distinguished Professor, Randall Prather, joins us for a two-part series focusing on innovation in swine genetics. We begin our conversation focusing on the basics of genetic engineering, and Randall shares examples of how this advanced technology could benefit the industry. Our conversation continues with Randall explaining that outside-the-box thinking has led to numerous medical advancements. From glow-in-the-dark pigs to heart transplants that grow, he discusses work that will leave you in awe. More in this area of research will be featured at K-State's Swine Profitability Conference, which will take place on Tuesday, February 7th. We end with K-State dairy specialist Mike Brouk discussing Bovine Leukemia Virus. He says that there are new tests that can help producers identify which animals to move out of the herd. That and more is coming up ahead on Agriculture Today. This is Agriculture Today. I am joined now by Randall Prather. He is from the University of Missouri, where he is a Curator's Distinguished Professor. And he's joining us today because he's going to be speaking at the upcoming Swine Profitability Conference here at K-State. And he's going to be talking specifically about genetically engineered swine for both agriculture and medicine. So, Randall, thank you so much, first and foremost, for joining us today. I am so excited for the conversation we're about to have. And I think this topic's going to be one that draws listeners in right from the start.
Hi, Samantha. I'm glad to be here.
Absolutely. So, like I mentioned, we're going to be talking about genetically engineered swine. So I guess the beginning of our conversation could kind of start with that. Genetic engineering, GMOs, the idea of CRISPR technology. There's a whole realm of things we could talk about, but we're going to talk about the basics, really.
So when we think about a cell, we need to kind of think about it as a factory. And the factory does a bunch of different things. The factory has a blueprints in it so that it can make a new factory. It brings in materials, supplies, whatever, into the factory to make the parts to make the new factory. It makes the tools to make the parts to make the new factory. And it has to self-assemble it into a new factory. Why don't you think about how complex that is? When we look at those blueprints, some of those pages actually of the blueprints, they only get used maybe a couple days in all of development. So there's some that are produced, some pages that are used during the period at which the embryo tells mom, hey, you're pregnant, don't cycle. And those pages are only used at that time. Well, what we can do is go in and modify those pages. So we can pull pages out. We can add pages from other species or other factories, or we can put in a new page so that the factory will have a new function. An example here are the omega-3 fatty acid pigs that we made. We went into C. elegans, which is a worm. We pulled out the page of the blueprints for converting omega-6 fatty acids to omega-3 fatty acids, and we put it in the pig. And now we have pigs making omega-3 fatty acids. So when you think about the things that we can do, we can go to other species to get pages, but we can change the page that we have. So let's take a piece of that factory, the hammer. Well, the hammer does a couple different things. I think most people think hammers as driving nails. Hammers also pull nails. So a lot of proteins in the cell are the same way. The protein's made up of a string of amino acids, and that string folds up into a three-dimensional shape, and shape gives you function. If we change the blueprints so that the string of amino acids is a little bit different, that will fold up into a different shape, and therefore you'll have a different function. So if we go back to the hammer, if you want that thing to hit nails harder, there's a couple things you can do. You can make the handle longer. Well, in the cell, if there's a protein there, we can change the shape of it. We can put a bump in the middle of the handle so it doesn't slip out of your hand as easily. We can change the shape of the head of the hammer. It's circular now. Maybe we can make it into a triangle. You want it to pull nails better? We can put longer claws on it. We can change the shape of that any way you want, as long as it's compatible with life. So now when we want to change the pig, change the amino acid sequence of any protein, of any part in that factory, and it has to self-assemble. That is so complex. Any part in that factory, we can potentially go in and change the letters of the genome that are basically the blueprints for that cell and change that protein and then change the function and possibly change the function of the cell.
Incredible. I told you beforehand when we were chatting before we went on air that this is giving me flashbacks to a molecular genetics class that I took in college. And I just thought it was so interesting because obviously this is a matter that's so complex. But it's incredible because we have the technology today to understand and to be so specific about the modifications that we are making. But sometimes we run into roadblocks with that technology, unfortunately. And there's a lot of different roadblocks, but it's inhibited a lot of growth. You mentioned heart-healthy pigs there a moment ago, talking about, omega-6 to omega-3s. But there's some other examples, too, that we can think of. You talked earlier about the PRRS virus with me.
Right. So the PRRS virus, I think the estimates kind of a decade ago are $660 million a year in North America, about 1.5 billion euros in Europe. That's $6 million a day in North America and Europe. That doesn't include Asia or South America. That's just North America and Europe. The vaccines don't work. They actually had this thing called antibody enhanced infectivity. And so the PRRS virus actually goes to the white blood cells and infects them. So if you have antibodies that recognize the PRRS virus, where does the antibody take that virus to process it? Right to the white blood cell, and it gets infected. So the vaccines don't work. There's not a good solution for this problem. Well, there's a molecule that sticks up on the cell surface. It's called CD163, and it has a bunch of repeat domains in it. And what we can do is change a few letters of a 3 billion letter genome and basically get rid of that protein so it's not made. Kind of like ripping the page out of the blueprints and throwing it away. So now that just doesn't get made. Well, since that's what the virus uses to infect the cell, the pigs are resistant. They don't exhibit any of the symptoms. You have wild-type pigs, pigs that don't have the modification, and pigs that had this gene knocked out or gotten rid of in the same pen, and you infect them all, and the wild-type pigs are coughing on the knockout pigs. The knockout pigs, they act like the virus isn't there. It affects the pig like it affects you and me. We don't get it.
Wow.
It's really pretty amazing. So we've done that for PRRS. We've also done it for TGE, different protein, aminopeptidase N. We knocked out the receptor for Seneca Valley virus. I don't know if you're familiar with that, but that presents itself as blisters on the coronary bands and on the nose. You get that, they shut you down until you prove it's not FMD. And so there's solutions to a lot of these viruses. Yeah, this has been really exciting to try to understand this.
Absolutely. And I think listeners tuning in are probably thinking, well, why don't we see this in industry right now? These are huge issues that we combat every day. They have to do with animal welfare, sustainability of our businesses. When we think about, you mentioned the dollars lost just with PRRS virus every single day, but also, food security for that matter. So why are we not seeing these things play out today in our industry?
Yeah, so the Food and Drug Administration looks at intentionally changing a letter of the genome in a pig or a mammal as a new drug. And so they regulate it as a new drug. Well, that costs an awful lot of money to get regulatory approval. That's the holdup. It's interesting because if we put things in perspective, at every fertilization, there's 1 to 200 random mutations that occur in the genome. Nobody regulates that. Nobody perceives that as a problem. Nobody as a health risk or anything. It's fine. You can take a pig and do what's called random mutagenesis, either with radiation or ENU-mediated mutagenesis, and do all kinds of mutations in the pig. We can eat those. Nobody regulates that. If you're working with fungi, the mushrooms, so they've already used the same technology that we've used to change a couple letters of a 3 billion letter genome in the pig to make them resistant to PRRS. They've used that exact same technology to knock out a gene in mushrooms so that they don't spoil on the shelf, so they have a longer shelf life. Actually, you're already eating those, and nobody has regulated that because nobody regulates fungi. But because it's an intentional mutation, and we know what that mutation is, we know what letters of the genome we changed, all of a sudden it's a new drug. FDA says they're science-based in everything they do. I'm not understanding how this is science-based. I'm not understanding how a piece of DNA is a drug.
So there's just a lot of barriers, it sounds like, in terms of getting this stuff implemented. Because, like you've said, this could be something that is a game changer not only for the swine industry, but there's plenty of other examples in other industries as well in agriculture that this could be beneficial for, right?
Well, so we talked a little bit about other genetic modifications we've done. Okay, so I can give a couple of cattle examples. One is mastitis. Almost for 2 decades, we've had the solution for mastitis, and it's introducing a new gene from another species that results in the staph A not being able to live in the mammary gland. We have the solution for this incredible problem in the dairy industry. It's an animal welfare issue. We have a solution for that. In the beef cattle industry, we have bovine respiratory virus. There is a solution for that You change 2 of those amino acids in a single protein, CD18, and you can make them so they're going to be resistant to bovine respiratory virus. We have solutions for this, but trying to get it through regulatory approval.
Lots of hoops to jump through. Yeah, absolutely. Our conversation is not ending here. We're cutting to a short break, but when we return, we'll be back with Dr. Randall Prather from the University of Missouri, where he serves as a Curator's Distinguished Professor. We've talked about the agricultural benefits of swine genetics and research in that area now. But when we return, we'll be discussing the medical improvements that have been made possible because of swine genetics research. Dr. Prather will be covering this topic at the upcoming 2023 KSU Swine Profitability Conference, which will be taking place Tuesday, February 7th. When we return, we'll include more details about that, but registration is still open. So if you're interested, simply search for the 2023 KSU Swine Profitability Conference to register. If you're listening to us on the radio and are afraid you're going to miss out on our next segment because you're reaching your destination or have something else that you're having to switch to quickly here, don't forget the Agriculture Today programming is also available as a podcast. Our podcast is easily accessible on all podcasting platforms, including our website, agtoday.net, as well as Spotify, Apple Podcasts, and more. You can simply search us on any podcasting platform, Agriculture Today, Kansas State, and we are more than likely to pop up, and you can follow us there. That way you can simply have us pop up in your regular feed on whatever platform you're using and listen to us more regularly that way. Once again, you can easily access that on agtoday.net or any podcasting platform by simply searching Agriculture Today, Kansas State. We're cutting to a short break now, but we'll be back with more ahead on Agriculture Today. You're tuned back into Agriculture Today. We are continuing our conversation now with Dr. Randall Prather from the University of Missouri. Well, Randall, I can't thank you enough for this conversation so far having to do with agriculture, but we haven't even gotten into what this means in the medical world, really, because that's a huge part of the work that you do at Missouri. And I just think that this is a world that a lot of people don't even think about sometimes when we think about swine research. And it's a huge part of our medical industry. So let's talk about some of the cases where we see swine genetics play such an important role in how medicine works today.
Yeah, so swine are really the optimal species for many human conditions where the other species just don't work. So a lot of research has been done on mice, for example. Unfortunately, they're not able to answer a lot of the important questions. So let's give a couple of examples. First one, cystic fibrosis. This is caused by a mutation in a gene called CFTR, and it pulls out a single amino acid. So that string of amino acids, there's one that's missing, and it's a chloride ion transporter. Well, you can knock that out in mice, and you can knock out the chloride ion transporter, but you still get all the symptoms with cystic fibrosis. So, for example, 15% of the people with cystic fibrosis have Meconium ileus. This is a mucus plug in the intestine. Can't pass any feces. You have to get it opened up for that individual to survive. There's a blockage of the gallbladder, blockage of the bile duct, blockage of the pancreatic duct. And in fact, if you know somebody that has CF, many times they have to take their pancreatic enzymes, their trypsin, chymotrypsin, orally with their meals. They also get liver lesions. The males will have a blocked or absent vas deferens. And after that, they get lung disease. I think most people think of CF as lung disease. Well, when we made that same mutation that occurs in people, in pigs, 100% of them had Meconium ileus, 100% of them had blocked bile duct, 100% of them had all the symptoms that people get. So now the physicians have something that they can go in invasively and monitor development and progression of disease in a way that just wasn't possible prior to this. And they've been able to come up with treatments and therapies. We collaborate with the University of Iowa on this project. And a few years, Mike Welsh came down. He's the leader of the program. Him and David Stoltz came and visited the lab. They took the lab out to lunch, and then they stood up and made a speech. They said, you guys have no idea the work that you do in the lab, at the lab bench, working at the microscope, making these embryos, caring for the pigs that we make, and then providing them to us to do research on has impacted the way we treat our patients and has improved their quality of life. And so, in that case, we're kind of a small cog in a great big wheel. And it's really satisfying to be part of that. Well, let me digress just a minute. We've made green pigs. So if you go to the aquarium and you see the jellyfish and they turn on the UV light, the jellyfish fluoresce. Well, so we've taken the page of the blueprints out of the jellyfish and modified it a little bit and put it in the pig. And now we have pigs that when you turn on the UV light, those cells fluoresce. So why would you want to do that? Well, people like to use them for tracking studies. So one example that I like to get is if you're studying retinal damage in humans, so damaging the back layer of the eye, the retina. You'll take a pig and you damage the retina to simulate what happens in people, and then you want to do a stem cell therapy or retinal progenitor cell therapy. You'll take these cells and you'll transfer them in the eye, and then you'll determine later that, yeah, it repaired the condition. The pig can now see and things look good. But is it the cells you transferred? Well, you can take that eye, put it on the microscope, turn on the UV light, the cells fluoresce, and oh, the cells that I transferred actually facilitated the repair and the treatment works. We distribute these pigs more than anything else to people around the country. So pigs go to Miami, pigs go to Washington State, pigs go to Columbia University in New York City. We send pigs everywhere. I don't know if you heard last summer about baby Owen. Baby Owen was a child that had a valve replaced in his heart. Well, the backstory is a few years before that, Conrad from University Medical School, South Carolina, came to us and he said, we have a problem when a baby's born and they need a new valve. We can put a new valve in, but the valves don't grow. So as the baby grows, we have to go back and do repeated surgeries. He says, I have this idea that if we take a bigger piece of the heart, in other words, doing a partial heart transplant that contains the tissue around the valves, that the valves will grow. And he said, I want to use your green pigs for this so we can see that those are the cells that grew. And so we sent him some of our green pigs, and he did pig-to-pig transplants. And we get this email back saying, here's the data. Nobody has ever seen this before. The valves grow.
Wow.
A month later, we get an email saying that the surgeons at Duke found out about it, and they had a baby born that needed new valves, and they were able to find a human donor, and they did this partial heart transplant, and it was a success. Maybe you saw it on the news this past summer. You should go back and look at it. But then right before Christmas, I got an email from my collaborator at Medical School of South Carolina, and it's a picture of baby Owen. And he said, baby Owen gets to celebrate his first Christmas because of the work that we've done. Isn't that exciting? I mean, we're impacting people's lives and improving their quality of life.
That's incredible. I literally have chills. I'm sure listeners do too as we're hearing this conversation because, like you said, this work is changing not only the way things are done in agriculture, obviously in our past conversation, but also how things are evolving for humans in the medical world. It's incredible. Well, if you watch the video of the story that came out of Duke, you better have your Kleenex handy. Yeah, tearjerker for sure. I mean, that's a family you've impacted, a child who will go on to hopefully grow to be a very strong and healthy adult and, have... And not have to have repeated surgeries. Wow. We talked about animal welfare before, quality of life for even humans even. Absolutely. Impacted. This research it's... I can't think of any other word other than incredible. Like, it makes my mind want to explode with how many possibilities are out there. And that's a huge part of what your discussion at the Swine Profitability Conference is going to center around, is that the options are really endless with this research, right? There's so much that if we just opened our minds to, that could be achieved through this work. So you want to come to graduate school? Well, I have my master's, but... I mean, I can't think of anyone that would attend this seminar, this conference, and not walk away being deeply moved by the work that you and others are doing at Missouri. And then obviously the work that's carrying on as a result of it. Like you said, a small cog in a big wheel, but I think you deserve a little bit more credit than just a small cog. We're a small cog in a big wheel. That's incredible. Especially for that project. Really, all we did was supply the pigs that we had for baby Owen. But still, like you said, the ripple effect of this research, it's limitless. You'll never truly know. Who would have thought green pigs? Green pigs, really? Yeah. And you'll be showing video of those pigs at the conference as well, which I got to see a clip of. And very cool. And it's, fun to see the switch of the lights on because just in broad daylight, they look like normal, healthy, happy pigs. So incredible. Well, Randall, I'm trying to think of, what else could we possibly say to listeners to encourage them to come to the Swine Profitability Conference, but I truly think this conversation was enough. And this isn't even all that you'll be sharing. There's going to be so much more that you're going to talk about at the conference. Well,
at the conference, and then they broke me into seminars on campus in classes and stuff. And so there's different things I'm talking about to different groups. So, there's a lot of neat things to talk about.
Absolutely. Yeah. So encouragement for those tuning in that have enjoyed this conversation just on Ag Today prior to the conference to make sure you're registering. The Swine Profitability Conference is coming up. I believe it's February 7th. Yes, Tuesday, February 7th here in Manhattan. And registration is only $50 prior to the event, and that can be done at any time prior. So those tuning in that are interested, please be sure to do that and check out more on this conference. We'll have all of the links to more information in the show notes of today's program, which can be found on agtoday.net as always. But Randall, thank you so much. I'm going to hopefully get some links from you about some of the incredible work still going on at Missouri that I'll link to below if people are interested in checking it out. But thank you so much for your time today. I truly am so appreciative.
Then you can think about a doctorate.
Yeah. We'll see. Once again, everyone, that was Randall Prather. He is from the University of Missouri, where he is a Curator's Distinguished Professor, speaking on genetically engineered swine for agriculture and medicine. Once again, Dr. Prather will be speaking at the upcoming 2023 KSU Swine Profitability Conference, which will be taking place Tuesday, February 7th in Manhattan, Kansas. While Dr. Prather's topic is obviously incredibly interesting, and there's still so much more that he's going to cover outside of the conversation we've just had at this conference. At the conference, you can also expect to hear from some more familiar faces like Dr. Glenn Tonzer. He's our K-State agricultural livestock economist, where he'll be talking about pork economics, assessing industry outlooks in a period of elevated uncertainty. You'll also hear from Dr. Paul Yeske. He's from the Swine Vet Center in St. Peter, Minnesota. He'll be talking about recent PRRS outbreaks and lessons learned. Once again, the Swine Profitability Conference will be focusing on the business side of pork production. Attendees will have a chance to learn ideas for improving their businesses from leading producers, veterinarians, and economists with close ties here to K-State. The conference is designed to help producers stay competitive in today's swine industry. So if that sounds like something you are interested in, be sure to register. The cost is $50, and you can do so up until the day of the conference, which is again Tuesday, February 7th. Again, this is an opportunity you're not going to want to miss out on. We'll be back with more shortly on Agriculture Today.
From Kansas State University, this is Agriculture Today. Along with Samantha Bennett, I'm Jeff Wickman. We end today's program with this week's milk lines. Bovine Leukemia Virus, or BLV, is an infectious disease that's been around for a long time. K-State dairy specialist Mike Brouk says that more than 50% of dairy cattle are impacted by this virus, and that a small percentage will develop tumors or other issues. However, he says there are new tests available that can help dairy producers identify which animals to move out of the herd.
Today I'd like to visit with our Kansas dairy producers about bovine leukemia virus, BLV. You know, we've been talking about this infectious disease for a long time, and, we go back to the 1960s, 1970s. It was not really very prevalent in our dairy herds, but across the United States today, more than 50% of our dairy cattle are actually impacted by this virus. Now, the difficulty with this virus has always been that a small percentage of these animals will actually develop tumors and other issues as they get older in age. However, you can go through and you can do testing in your herd, and you've been able to do this for a long time, and you can determine which animals are positive or negative based on an ELISA test. While it's important to maybe do that in your herd. It's also difficult to walk in and cut your herd in half just because they are positive when they're not showing any other signs or issues. And a small percentage of those animals will develop those types of issues later in life. So maybe there's some things we can look at a little bit differently today. There's some new tests that are available that may help you identify which animals you should be moving out of your herd. Just because she's BLV positive doesn't necessarily mean that you want to move her out of your herd. However, if you visit with your veterinarian, you'll find that there's a new test that's available. It's called a BLV proviral load test. So what this does is this is a laboratory test that we can now do to basically measure the amount of BLV provirus that's found in the blood. As we get higher and higher levels, it's more likely that these animals will develop issues. So we could be moving them out of our herd. The other thing it helps us do is it helps us control the level of BLV in our herds. Those animals that have a high provirus load are also those animals that are shedding a higher level of the virus. So they're more contagious, if you will, to other animals around them. In Europe, over the last couple of decades, they've been able to actually eliminate this pathogen from their livestock by using this type of testing. So as we look at things that we might want to do in our dairy herds moving down the road, we might want to look at a way to eliminate or at least lessen the percentage of animals in our herd that test positive for BLV. And the most efficient way to do that would be using this new proviral load test, because it identifies the animals that are high shedders in our herd and also animals that would be more likely to develop issues later in life as opposed to just an ELISA positive test for BLV. This is Mike Brouk with K-State Research and Extension, encouraging our dairy producers to visit with their herd veterinarian about new ways to control BLV in their herd.
Thanks, Mike. And while we have a moment, this reminder about the 2023 Women Managing the Farm Conference being held February 15th through the 17th in Manhattan. From ag advocacy at large and small scales to the impact of military aggression on global food security to mental health, the conference offers women the chance to explore how to grow their operations, their communities, and themselves. Presentations will cover a variety of topics, including farm finances, agricultural and estate law, crop production and marketing, management, farm succession, direct marketing foods in Kansas, and much more. For more information, including costs and registration, visit womenmanagingthefarm.com or agmanager.info. That's womenmanagingthefarm.com or agmanager.info. And that'll do it for this edition of Agriculture Today. For Samantha Bennett, I'm Jeff Wickman. This is the K-State Radio Network.